Hello, everybody, and welcome to another episode of How to Live to 200. On today's episode, I had the opportunity to speak with Ivan Liachko, the co-founder and CEO of another University of Washington spinout, Phase Genomics. Phase has developed an entirely new genomic discovery method that, among other things, can help lead to longer lives and new drug developments. Ivan has his Ph.D. in genetics and has been working on genetics or genomics research for over 20 years. In this episode, we explain and discuss the importance of genomic sequencing and why he studied Lil Bub's microbiome. You know, that cat of internet meme fame with the big eyes and the cute little tongue? And how he found entirely new organisms in cat poop. We also discuss what he thinks might be an effective alternative to parabiosis, or transfusing older people with younger people's blood. Yeah, I know. We're still working on getting a guest to talk in detail about that strategy. Like past guest Alex Jiao of Silene Biotech or Leen Kawas from M3 Biotech, I love to talk to scientific founders who are taking these important problems into their own hands and building companies around them. And with more venture capital flowing into the longevity startups than ever, I'm excited to talk to more founders in this space. Finally, while he didn't mention it by name on the show, how cool is it to work on a product called a Proximeta High-C metagenomic deconvolution platform? And I just wanted to say it once before we got into the episode. And now, this is how to live to 200. Ivan, thanks a lot for coming, being on the show today.
My pleasure to be here. Thanks for having me.
Tell me a little bit about Lil Bub.
Lil Bub is an adorable celebrity cat. She was born with a number of developmental conditions that make her extremely cute, but she's also sweet, which may or may not be.
What are the developmental conditions that Lil Bub has?
She's little. She is like a perma kitten, as they say. Like, she's tiny and she has giant eyeballs. She has, like, her tongue sticks out all the time, and a lot of that has to do with, like, she's missing teeth on the bottom and her jaw isn't growing all the way. So it's like, but the net effect is very cute. And she's also, like, she just has a nice personality, and her owner is really cool. And they, like, they have, like, a little talk show. They've written, like, books, and a lot of the proceeds of all that stuff goes to charities for helping animals, that sort of thing. But, we were doing, we do a lot of microbiome work. And I met with Holly Gans, who runs an animal microbiome startup called Animal Biome. And we were just chatting about projects, and we said, like, what would be a cool thing to do? And she said, I have a sample from this famous celebrity cat. And Lil Bub apparently also has had her genome sequenced, like the cat's genome. And so we said, well, if they did that, then let's do her poop. And so we sequenced Lil Bub's fecal sample, and we used our special technology on it, and we discovered a whole bunch of new bacteria, and then talked about it on Twitter. Did a little Twitter contest, had somebody, a winner, had people submit their cats, and then we picked somebody. Well, not picked, randomly selected a winner, and now we're going to do her cat sample.
And what did you learn from that's now going to be applied in a scientific sort of way, other than it being fun?
Yeah, well, so one of the things that people may or may not be aware of in the microbiome space is that it's like the wild, wild west out there. We know nothing, right? So people know sometimes little statistics like sometimes when I talk about it, I ask people, are you think of yourself as mostly human, but actually the vast majority of life in you is not human, right? So most of the cells in your body are not human; they're microbial. And if you count not by cell number but by like number of genes, if you just take all the DNA and look at all the genes, you're outnumbered by microbes by like orders of magnitude, like 1,000 to 1 or something, 100 to 1.
Yeah, I've heard 150 times to 1 in terms of
DNA.
There's sort of controversy to how much and whatever, but yeah, significant, right? You're not like, it's not like you're a human being who happens to have a few bacteria hanging around. You're more like a walking universe. You're like a scaffold for these things, and humans, the human gut microbiome has been studied a lot. Like that's by far the most studied niche. The moment you get out of the human gut, you're in like the land of unknown. So you basically, if you go to soil or water or anything, if you actually look at what lives in these environments, like 99 whatever something like that % of all the microbes you find will have never been identified by science before. And that's one of the points we were making. Like Lil Bub's poop, she had some microbes that were known, largely because, so there's this idea that pets and their owners share microbes. It's slightly gross, but it's probably true,
But that's more environmental, right? They both live in the same environment, therefore they're likely to at least share some of the same microbes that might be in environment 1 versus environment 2.
Totally. And so, but there were still a bunch of unknown microbes there as well. And so kind of the goal of the project was not so much necessarily to discover some specific thing about Lil Bub's environment, but to highlight the fact that any microbiome sample, even human ones, every time you process one of these samples, just new stuff comes out all the time. And it's more dramatic if you go outside of guts.
Because it's less studied?
Partly because it's less studied, partly because it's more complex. So, we by the time this airs, this paper would be out. But basically, we took a sample from sewage, from wastewater that we got from a collaborator, and there we pulled out about 1,000 different microbes that have not been studied, have never been identified before. The same thing, we've done work with a group in Scotland on cattle rumen. So that's like the inside of the guts of cows. Again, hundreds of novel things. And so soil is kind of known to be like the super complex ecosystem. And so human guts tend to be a little bit less complex, and they've been studied to death. And so so as soon as you move out...
But studied to death, but still relatively early. So the work being done in gut microbiome, what's in there, how it can be changed, affected, is still relatively early.
Oh, yeah. And I think that highlights just the sheer complexity of this. Because, it's hard enough to sequence one genome. Try sequencing 1,000 genomes when they're all mixed together, right? And that's kind of actually the central kind of point of what Phase does, what we do. Like that's what our technology solves to a large extent.
How is your technology novel?
So what our technology does is it basically, it has a couple things you can do with it. But in the microbiome space, what our technology does is it tells you which sequences of DNA started out in the same cells before the DNA was sort of broken up. So you can essentially, the big problem with the microbiome, with sequencing a microbiome sample is, let's say you have 1,000 microbes, they all are living together. You purify their DNA to get sequenced, and the way that DNA sequencing works is it basically shreds all the DNA into tiny little pieces, and then you sequence the little pieces, you read the pieces. Now imagine you did that when you have a mixed community. You lose the information of which pieces came out of which organism. Sometimes the metaphor I use is jigsaw puzzles, right? So imagine you have a huge jigsaw puzzle of millions of pieces, and now you mixed 1,000 of these together. You dumped all the pieces together, you mixed them up. Now try to solve these jigsaw puzzles. You don't even know which ones go where. And that's the problem. That's why it's so hard to do this. And what our technology does is it tells us which sequences started out in the same cells before the DNA was all purified. And so we are able to then, after the sequencing is done, separate all those genomes out. We can tell these sequences came from one organism, these sequences came from another organism. And so we are essentially able to put genomes together for hundreds of bugs completely de novo. De novo meaning without, like brand new, like without having to be informed by a database or kind of without taking guesses.
Why does this matter? Especially as we think about humans or animals living a long time. Why does this type of technology, this kind of understanding matter?
Sure. Well, I mean, one sort of high level answer is, basically life is information, right? Like we all started out as one cell. That cell has some DNA in it. The DNA provides the instructions for how to build a person. Everything else has a genome too. That's sort of like is the foundation of all life. And so what genomics seeks to do, and genetics and other fields, is to try to extract as much of that information as possible and to actually understand it, to put it into context. And so to some extent, it's basically a race from a low information state to a high information state. Because presumably, if we can figure out everything about how a certain organism works in terms of sort of the instructions that are used for developing it, you can potentially extend its lifespan, you can cure diseases, you can do all sorts of stuff, right? Somebody just published that they CRISPR'd a baby in China. So you can start, there's sort of good things, there's some scary stuff potentially in the future, but it essentially gives you the power to take control of the situation in a way that you can't if you don't know. So for example, in the microbiome space, right, there is this amazing kind of revolutionary cure for colitis, which is called FMT, right? Fecal microbiome transplant. And this is like a treatment that works. It's like 90% effective for a disease that for decades was thought to be uncurable. Like that's it. And then, but what a fecal microbiome transplant is you take poop from a healthy person, you put it into a sick person, and the sick person is magically healthy now. And nobody really knows why. There's like patterns they know, they think it has to do with biodiversity and that kind of thing. But nobody really knows what the causative factor is, right? So it's like, it's basically medieval type medicine. You're like, this seems to work, let's keep doing that, right? What if we actually understood what was going on, right? A lot of longevity questions and mortality questions, right? Because one way to live longer is just to not die for longer. Is how do we prevent diseases? What are the genetic factors that cause a person to live longer or to live shorter? There used to be kind of sort of the intuitive model for how aging works is sort of like the car model, where it's like you buy a car, it's shiny, it's brand new, and then 15 years later it's all like rusty and beat up and stuff's breaking down, and and that's kind of like how we age. But that's not what's science is pretty clear that there's specific genetic instructions that your body is doing specific biological things that make you age. You're not just breaking down. There are specific signals that cause that to happen. And so in a lot of model organisms and other sort of areas, you can actually extend the lifespan of whatever it is you're working on. You can take one of the main models for this is these little tiny worms called C. elegans. It's a tiny little microscopic worm that if you give it certain mutations, if you put certain mutations into it, you can make it live like 3 times longer than it normally does. And it's not just surviving longer, it's actually it seems to be healthier proportionally. It's almost like you stretched its lifespan. You didn't just add a bunch of years on. But then if you put them in the wild, they actually die. They're less fit in the quote unquote the wild. So
has that C. elegans work been applied to mice or mice models or even more complex sort of animals?
People try all the time. They've done it actually with fecal microbiome transplants. They've shown that you can extend lifespans of these things by doing that sort of stuff. It's a little bit hard to do that in humans because remember, this is actually aging is a hard area of research, because think about what an experiment entails. You do something to an individual and then you say, all right, let's see if this person or this thing is going to live twice as long as it's supposed to, right? And so you can't really do it on people because you have to wait another 50 years to know the answer. That's why they do it on model organisms.
Can you do FMTs in mice?
Yeah, they've done a ton of it.
And that FMT in mice, has that also extended lifespan? So there's disease prevention, and then there may also be life extension.
Yeah, FMTs do all sorts of amazing things in mice. There's they prevent diabetes. They do all sorts of things. Lifespan extension. There was a paper in in fish, where you can do a fecal transplant and extend their lifespan by 40% or something like that.
Is a young microbiome significantly different than an older microbiome?
So what's interesting is sometimes microbiome people...
It's like parabiosis, but for microbiome.
Right, so...
We're going to get young people's poop as well as young people's blood to stay younger.
Well, so but the difference is that there's not really much evidence that like using their blood will extend your lifespan, but fecal stuff, there is actually a good amount of evidence for that kind of stuff. So, you think about like ancient medieval rulers who would like bathe in the blood of young people to stay young, like they were bathing in the wrong thing. But again, we have no idea why. Like nobody knows why. And the way that they think often about the microbiome is kind of like a forest. Like when you're young, you're like there's like seedlings, like the things sort of develop and they grow and progress, and your microbiome matures over time, and then its complexity goes up, and then when you're old, it kind of starts coming down again. And so there's definitely sort of developmental stages of your microbiome with age. There's a lot of these kind of chicken and egg questions. Is it your microbiome changing that's causing you to age and have certain diseases? Or is it the other way around? You're aging, you become more susceptible to like inflammation or something like that, and that your microbiome like can't deal with it or deals with it in a way that doesn't help you or something along those lines. So that's really the problem. The problem is that you suddenly have to do experiments that are so complex, that have so many variables, you just don't even know where to start. Yeah. And so back to your original question of sort of why does it matter, right? If we want to solve these problems, we're going to need more information. We have to start figuring it out. The most common technology used in the microbiome space, for example, is this thing called 16S sequencing, where... Is
this what someone like Ubiome may use, or more commonly consumer-oriented microbiome or gut microbiome testing scenarios?
Yeah, so a lot of those...
16S.
16S is definitely the most widely used technology. It is the most inexpensive. And what it does is you sequence or you somehow identify one piece of a bacterial genome. So like there's this gene called 16S. Every bacteria has it. It's one of these super required things for life. But it changes. There are parts of that gene that don't have to be the same. They can change. And then there are parts that cannot change or the thing won't work. And so if you were to sequence it from two different bacteria, you can say that they have the same the regions that are called what is called constrained, meaning they're like the same, they can't change, they can't put mutations in it. And then there are regions where if you put mutations in it, nothing really happens. And so you can use these variable regions to give you information about sort of where this organism came from on the sort of the evolutionary tree. You can say kind of what species is it from, like roughly, what like genus, what clade is it in, right? The problem is it doesn't tell you anything about the rest of its genome. You're just sequencing this one little piece of the genome to kind of identify which branch of the tree of life it's coming from. It doesn't actually tell you anything about what its genes are. And it also requires you to know what the different options are. Like if something was truly new and you got a 16S sequence from it, you would have no idea where this thing goes.
So given what you know about the gut microbiome and the sequencing, do you feel like there's value for a consumer to do something like Ubiome?
Oh, sure. There's value. There's value in the way that there's value for... Is there value to do 23andMe? It's mostly... Yeah, it's mostly kind of, for fun. It's educational. For me, for example, 23andMe was important because I found out that I'm 12.5% Jewish, right? And that's something so I'm an immigrant from Eastern Europe, and there were kind of like stories that there was some immigration out of Germany in the 30s. My great granddad moved, and we never really knew that was true or not, but it kind of makes sense, right? So you can discover a lot of really interesting things about yourself doing that way. Whether or not it's useful, nobody's really figured out what to do about it, right? There are certain clinically actionable things, but unless you have some glaring problem or some glaring kind of issue, it's mostly at this point kind of for fun.
We're funding the science.
Yeah.
So give us sort of a simple primer on genetics, genomics, sequencing, testing. Like start with something that seems like the 8th grade version and then quickly gets into the state of the art.
Sure.
In 2 minutes or less.
I'm going to use a lot of metaphors. So one way to think about a genome is like, think of it as a book, right? So a book, it's humongous. It has a bazillion words in it. And then I gave it to you and I said, TA, can you read this book for me? But instead of giving you a book, I ripped it into pieces, each piece having 1 or 2 words in it. And I gave you this mountain of word pieces. And you would look at them and you would say, I read this word, and then you throw it away and you say, I read this one. And you would read all the pieces. And then you would say, I read your book, right? And clearly, like, that is true on one level. You read all the parts of the book, but you didn't really read the book. And that's how genome sequencing works today. So your genome is, think of it as sort of instead of kind of a book format, you know those old telegraphs that would like shoot out like a ribbon, like a little paper ribbon and go beep, 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 and it would put like Morse code on it? That's kind of what your genome is. It's a super long, it's like miles and miles long version of those. But instead of dots and dashes like Morse code style, you have A's, T's, C's, and G's. That's all it is. And now imagine what I just told you with the book. Like, you ripped it into like tiny little pieces, and then you started looking at the pieces and trying to divine sort of what's going on. It's extremely difficult, right? But that's how all genome sequencing works today. You take extremely long DNA molecules, you break them into little pieces, and then you read those pieces, and you try to get computers to put the pieces together based on like the overlaps between the different sequences. So you find like unique regions, you might be able to splice sentences together, that kind of thing. And so the main sequencing technologies today are provided by companies. So Illumina is the biggest sequencing company. Then there are Specific Biosciences and Oxford Nanopore. And Illumina, what Illumina's platform does, it is by far the most used. It shreds your DNA into little like 100 to 300 base pair pieces, and it just gives you those. And so, but you get millions of them. So you'll get like hundreds of millions of these short reads. And then you have to try to stick them together. Companies like Pacific Biosciences and Oxford Nanopore, they're long read technologies. So they give you fewer reads, but they're way longer. So instead of reading 300 letters at a time, you're reading 10,000, 50,000, 100,000 letters at a time.
Is this like a chemical process that they're going through?
So there's different chemistry. So the way that Illumina sequencing works is you basically you attach lots and lots of individual DNA molecules to like a little chip, and then you start copying the DNA molecule. And while you're copying it, every time it adds on another letter, it flashes a certain color. And you have optics that look at it from above, and they can see which colors get added sequentially, and that's how you know like it's an A and a C and then a T. PacBios uses this interesting. It takes a protein which is normally used to copy DNA, called polymerase, and it sticks it at the bottom of a little nano well that's only big enough for one DNA molecule to fit in. And so then the one DNA molecule goes in there, it sticks to the protein starts reading it. And as it reads it, with every letter, it gives off a little electron signal, and you can sort of read it that way. That's called single molecule real-time sequencing. And then Oxford uses what's called nanopore sequencing. You have a protein that's like a little sausage. It's like a little, it's called a nanochannel or a nanopore. It is exactly what it sounds like. It's a tiny pore. And DNA can go through the middle of that pore. And as it pulls through, whichever letters start passing through the nanopore, it gives a little bit of a different electrical resistance. So you pass electricity, an electrical field through these nanopores, and they will like change. They'll go up or down, depending on which letter is going through the pore at that moment. And so you just do that, and you're kind of reading the ticks. And so nanopore sequencing is very popular now because it produces the longest sequences, the longest reads, and it also is less costly to some extent. But then again, it's also prone to a lot of mistakes because, like, if you for technical reasons. So certain sequences it has a hard time with. And so do you go for the higher accuracy short read sequencing, or do you go for the lower accuracy long read sequencing? You can imagine it depends on what you're trying to do.
If somebody were interested in either a critical health condition or longevity or both, what would you recommend that they try to do for themselves? So a 23andMe or a gut microbiome, or go to some place that's going to do one of the tests or use one of the technologies you just talked about?
So longevity is hard to predict these days.
I have a family history of colon cancer. Okay, so that may be something I want to pay attention to, and I want to sort of do some genetic testing related to that to see whether that might have been diet related or genetically oriented.
Yeah, so if you have a thing that you're specific, if you know what you're looking for, if you have a condition, I want to figure out if I'm at risk for having that condition. There's a number of diagnostic companies that will kind of give you tests for that. They will sequence you. They will look at your different alleles. 23andMe may or may not be helpful. That might be a little bit too kind of low pass, but sort of low resolution. But that's pretty commonly done sort of in the professional and medical sort of arena. There are things like aging, though. Nobody knows. The big problem with genomics is, let's say we sequence all these genomes, we still don't know what stuff does. That's a much harder problem. It's easier to say, everyone who has this particular disease has this one mutation. Therefore, if we find that you have that mutation, we'll tell you and we'll say, hey, watch out, you might be at risk for this.
Right. We're more directional as opposed to directive at this point. Right. There's high correlation for certain genetic variants than there is specific. Every single person that has this variant has this outcome. Right. But that's more useful than where we might have been 10 years ago, which is we didn't even know what potentially it was correlated to. A BRCA gene or APO gene or.
And again, it's all going from sort of a lower information state to a higher information state. As we learn more and more and more, right, we can make better decisions, be more informed. And the technology is there to kind of assist that and help us extract more information.
What's the difference between the genome and the metagenome?
Sure. Well, is the word meta. And the meta...
4 letters.
Right.
I got it.
Yeah. But that's actually kind of the difference is that when you have a metagenome, it is a genome of the unknown. You don't know if it's one genome, if it's multiple genomes, if it's 1,000 bacteria, all of their genomes got mixed together. Like when you're sequencing somebody's genome, that's like one person, one organism, one species, right? Metagenome is kind of, I don't know what's in there. It's like I'm calling it one thing, but I know that it consists of many different things. I just don't know how to tell them apart.
How do you think this translates, your technology, how does this translate into whether it's human life or healthcare or whatever? What's the first sort of application?
Well, the most obvious thing, at least in the antibiotic resistance world, is as these bugs become more and more prevalent, which they are, killing them is going to become harder and harder. So more and more effort will be focused on tracking them and detecting them before they hit us. And so just developing a good assay for detecting antibiotic resistance before it kind of comes into play, being able to trace it, right? So say there's an outbreak, where is it coming from? That sort of stuff. Another element is just discovery, right? So a lot of what we do is we just discover new stuff by the gobs, right? And so we discover hundreds of new microbes sometimes from a single sample, and just accumulating that body of knowledge, you can then turn it and use it for all sorts of like informing drug discovery efforts, informing diagnostic efforts, trying to say like, which bacteria is causing some effect, which bacteria has which genes in it, so we can try to understand sort of how its how its metabolic processes can be sort of turned to our advantage.
What do you think is going to be commonplace 5 years from now that seems like science fiction today?
Honestly, I'm a big fan of stem cells, stem cell technology. I think it's I think it's going to go pretty far. I mean, I think obviously, like sequencing, DNA sequencing, it's what I work on, and we see that becoming very prevalent. We can see the rapid growth and the adoption of that technology, and so that's kind of an obvious answer. But I think aside from that, I think there's going to be a lot more targeted therapies, a lot more stem cell stuff.
Where can people find out more about you or Phase Genomics online?
Well, I mean, the number one way to get information on us is to visit our website at phasegenomics.com. We have both sort of a long publication section for people who are really interested in kind of going deep. We also have blogs, we have videos, we have how it works kind of sections. And so we can do that. Twitter at Phase Genomics.
That's great. Thanks a lot for joining us today, and exciting sort of future you're putting in place.
Thanks for having me.
You've been listening to another episode of How to Live to 200. Thank you so much for joining me and exploring this world together. I get a ton of help from the L200 crew that includes Lauren Krayinsky and Kevin Kirkpatrick. The theme music is composed by Emmett McCann. Yes, that's my nephew. You can learn more about this and other episodes at our website, livingto200.com, or find us on Twitter or Instagram at howtolivето200, where we post lots of photos of cool things. It's early days for this podcast, so we would appreciate any and all comments or telling a friend or two about what we're doing over here. It might be irresponsible for you to keep it a secret. Until next time, eat right, get lots of sleep, keep good numbers, and be looking around the corner for the next big breakthrough. If we're going to live a long time, we better do it well.